And welcome back to the podcast, guys. Back with another episode on this here podcast. And we're back with Chris again. Chris,
Hey, guys.
the wave. He's back. This is our 7th podcast this week. A lot of work.
Yeah.
A lot of editing, a lot of work. But we're making it work, guys. Just for you guys. And we got a new one. Kind of scientific.
Yeah.
Kind of cool.
So strap in, guys. It's gonna get complex.
Buckle up, if you will. And don't let those straps unbuckle, because this is going to be a wild ride. Started off though, look at this guy. Look at him. So cute. His name's Heinz. It's my dog. It's his birthday today. Just realized the entire science class is going to be seeing this video.
Say happy birthday, guys.
Yeah, happy birthday.
Say happy
everyone.
birthday.
Happy birthday. If you don't say happy birthday, I'm watching you right now.
We're in the same class. Yeah.
Alright, go ahead.
All right. Speaking about birthdays, let's talk about birth. Sperm and egg cells, to be exact. How are they made? Well, meiosis. Let's get into it.
And Gavin cannot be here.
Oh,
He
that
had
is
baseball.
true. All right. Lab partner.
Too good for us, I guess. Right, Gavin?
Big
right?
Jason.
Right? Anyways, what the heck is the purpose of meiosis, right, guys? You might be wondering that right now.
I am.
Well, have you ever wondered how you got here? You yourself got here? Well, the answer to this question is, guess what? Right in that word, meiosis. Pretty cool. It's how eukaryotic cells reproduce sexually. Now, this is solely for eukaryotic cells, and sexual reproducers go through this process also. And in this process, in meiosis 1, the cell goes from a diploid to haploid cells. A diploid organism, of course, to haploid cells. And then in meiosis 2, goes from haploids to more haploids. So let's get into it.
All right. So, meiosis is made of 2 parts. So let's get into meiosis 1. It starts out in interphase 1. The cell grows and gets ready for the next few stages, which happen to be the same as mitosis. You got prophase, metaphase, anaphase, and telophase. So let's get into prophase 1. Chromatin condense to form visible chromosomes. Spindle fibers begin to appear. All right. In metaphase 1, spindle fibers connect to each sister chromatid, and the chromosomes align in the equatorial plane. In anaphase 1, spindle fibers pull sister chromatids. Spindle fibers pull. Sister chromatids are sister chromosomes at this point. In telophase 1, spindle fibers disintegrate, chromosomes decoil to form chromatids, and the nuclear envelopes form before cytokinesis, forming 2 haploid daughter cells.
And of course, in interphase 1, at this point, guys, we got G1, G2, and the S phase happening. None in that order. It goes G, S, G2. And the anaphase 1 gets a little choppy here. It's a little confusing. The sister chromatids are sister chromosomes at this point in anaphase when they get separated apart. Just to clear that up. Let's get moving on to meiosis 2. It begins with interkinesis or interphase 2. And this differs from interphase 1 because of no S phase. The DNA is already present. Pretty cool. Then goes in prophase 2, where the chromatin condenses to form visible chromosomes. Spindle fibers begin to appear. And this repeats again. Metaphase happens again. Anaphase happens again. Same exact thing. And telophase happens again. And the nuclear envelope forms before cytokinesis. And this forms 4 haploid daughter cells. We're back in meiosis, whatever you want to call it. 1, 2 haploid daughter cells are formed by cytokinesis. And moving in, we got a picture here.
Nice picture.
Chris, you want to explain it to us?
Sure. So, as you can see, up here, starts out in interphase, right? Yeah? Normal interphase. Goes in, the DNA gets replicated. Replicated.
It's okay.
Then we go through meiosis 1. The diploid cells get split into 2 haploid cells, as you can see there. And I tried to make it obvious, but it didn't work out. Mr. Rollins didn't have the materials necessary.
What you did? I continue. Continue.
So the chromosomes get split equally, or should be at least, into 2 almost copied cells, daughter haploid cells. Then from meiosis 1, they go into meiosis 2, which each cell then splits into 2 more haploid cells. And those chromosomes get torn into 2 chromatids, all right? That's important. That makes each and every last haploid cell very unique in its own way.
Who's that cute guy in the corner of the screen right there?
His name
I
is
think Oh, it's Jeremy.
Jeremy Flickinger?
All right.
No. Yeah, Flickinger. Jeremy
So,
Flickinger.
all right. What the heck's the difference between Meiosis 1 and 2, Chris? Tell me.
Okay. So, well, the first main difference is that Meiosis 2 starts with 2 haploid parent cells and ends with 4 haploid daughter cells, maintaining the number of chromosomes in each cell, whereas Meiosis 1 starts with 1 diploid parent and creates 2 haploid daughter cells.
And the chromosomes in that doubled in Meiosis 1.
Alright. Also, in Meiosis 1, crossing over occurs, when Meiosis 2 does not happen. Alright? Also, the amount of time, M1 is a little bit longer.
Crossing over. Chris, you want to take it?
Sure, why not? I love this topic. Crossing over occurs in prophase 1. As we said, it only occurs in Meiosis 1. Chromosomes switch some genetic code with each other. So they say some chromosomes, they get compact together. They may switch around some DNA like that.
You're crazy.
And even though it's the start of the process, it makes the gametes at the end of the process very unique.
Cool. N versus 2N cells. Now, N represents the number of complete copies of DNA a cell has, haploid. An example are the sperm, the egg, individually. 2N is the combination of 2 of these N cells, and they create a new cell with DNA from each N, and this forms a diploid. The fertilization of the sperm and egg cell is one of the key examples of this, and any other body cell is another example. Here's a picture of it. I'll explain it to you guys. So we got you got your traits right here.
Yeah.
You got AA, which could represent
Eye color, hair color, even. I don't know. I'm
and
not that.
FW, FR, which could represent fur.
Yeah.
Fur white, fur red. All right, so you got one of this trait in here and one of this trait in the sperm cell, and then it combines with one of the other traits in here with another trait in this, and that's in the egg cell, and then they add up. Each of these is an N, and each of them is a haploid. They add up, they go into a diploid, and the traits come back together, and this is 2N, is what's happening in this picture. It's basically N versus 2N right there. Pretty well described, I hope. And now we're going to go into non-disjunction. Non-disjunction in meiosis is an issue that occurs in the separation of chromosomes with gametes. When the chromosome separates, it could have too much or not enough of a chromosome, and it's an uneven separation of chromosomes. And this occurs during anaphase of either meiosis 1, or it could occur in meiosis 2.
Alright, so some consequences of non-disjunction. There are 2 main issues that emerge from non-disjunction. The first one is trisomy 21, which is Down syndrome. It occurs when one of the haploid cells takes one too many chromatids from the other cell during interkinesis. The second one is monosomy X, which is Turner syndrome.
And moving on. And how variety is accomplished in gametes? Chris, go ahead, take it away, man.
You got it, bud. So, first off, crossing over, we already went over that. That's when chromosomes exchange genetic code with each other. There's random segregation, which chromosomes can go into each gamete at random chances. So, one chromosome can go into the 4th gamete, or the 1st, or the 2nd, or 3rd. It's all random.
Rando, we got a squad rando.
It's almost as random as dice.
That's a horrible comparison, actually. You know what? Just keep talking, buddy. Keep talking. Independent assortment.
Bro, really? Okay. Each chromatid has an equal chance of getting either allele, as we went over in our genetics chapter. So, obviously each trait can have up to 2 alleles. Has to have at least 2 alleles. It could have polyinheritance, which could mean many alleles. But for normal circumstances, let's just say there's 2. Each allele can go into either chromosome. So each allele can go into the 1st gamete, the 2nd. It's all the same. It's like dice.
Cool. And folks, that's all we got for you guys. I hope you guys know a little more about meiosis how you got here, and how this cute guy got here. And next podcast, we're gonna be trying every single Ben and Jerry's ice cream flavor, and then the one after that, trying every type of lunch meat there is on the market.
Speaking
Catch
of
you guys
which,
later.
Mr. Rollins.
All right, stop.